Well, back in school, you may have been told that human egg cells are made by the shuffling of your parents' genetic material. But the full story is more complicated. Because chromosomes from the mother and father are similar but not identical, mismatches occur when they line up for genetic exchange.
In a new study, Taiwan researchers discovered how a protein called the DMC1 works to mediate those mismatches to achieve functional new DNA. Their findings were published in the January edition of the journal Nature Communication.
When egg and sperm cells are produced, chromosomes from the mother and father exchange bits of genetic information to form a new combination of DNA. It's a process that's still not completely understood. One mystery for scientists is how a perfect new chromosome can be created, even when the parental pairs are not perfectly identical. The key to the answer is an enzyme called DMC1.
It's already known that the DMC1 can tolerate mismatch and for genetic exchange, but they don't know how this enzyme can achieve this task. So to solve this question, we combine the expert from the academic Seneca. They use the cryo-EM, this tool, to get the high resolution protein DNA structures.
Scientists know that DMC1 helps the new DNA tolerate imperfect pairings of parental sequences. To get more insight on how DMC1 does it, they used cryogenic electron microscopy. They took samples and cooled them down to cryogenic temperatures before studying them using electron microscopy. With this method, scientists were able to study the DMC1 enzyme at molecular resolution.
Okay, so being able to see that at atomic resolution, we can identify some key residue in the space. For example, we identified two small amino acids, glycine and purine, insertion into the DNA pairs. So that small residue has extra space. So when DNA pair is mismatched, they need more space. Okay, so they've been able to use that extra space.
The team found that DMC1 enzyme has a unique structure that binds to the DNA strand, creating an extra space that increases flexibility during pairing. Researchers say that mutations of the DMC1 can be an explanation for some forms of infertility.
If we know this process, as I just mentioned, that because this process is important for meiosis, okay, it's how you get a sperm, you get the eggs, okay, for the reproduction, okay. So you if understand this basic of this process, later you can see if some patient have fertility issues. We can predict or we can look at where this gene in the patient is, where has any mutation in those amino acid residue. So in that case, you can predict maybe this is the cause for the fertility issue.
The Academia Sinica established its cutting-edge cryogenic electron microscopy facility 2 years ago. It's currently using the facility to investigate COVID spike proteins.
Worldwide right now, in order to be able to see the spike protein and coronavirus, the key technology still is cryo-EM. So this is the first high-end cryo-EM facility in Taiwan. So many of researchers in Taiwan, if they want to see their spike protein or virus, they will send over here. So we already helped some research to solve atomic resolution spike protein of the coronavirus, which already published last year.
With COVID spreading and evolving fast, scientists are scrambling to study the structure and function of its spike proteins. The Academia Sinica's cryo-EM facility stands on the front line of researchers' war against the pandemic. Formosa News, Stephanie Yang, Liao Wenpu in Taipei.